So this presentation is a semester project in the course Applications and Technologies of Synthetic Biology, MCB 6937, which is part of the Microbiology and Cell Science Department at the University of Florida at Gainesville. And the title of the paper that Group 2 selected was Synthetic Cell Lines for Inducible Packaging of Influenza A Virus. My name is David Kapp, and I'm going to be talking about the background and getting into some of the design and key results. In terms of the background of the paper, the main motivation is that seasonal influenza causes significant morbidity and mortality worldwide, with over 1 billion infections, between 3 and 5 million cases with severe illness causing hospitalization, and up to 650,000 deaths per year. In addition, in the U.S. alone, we spend $10 billion in medical costs, with over $87 billion in total economic burden when you consider the lost time and wages of those that fall ill from the disease and those that pass away. But vaccines are still the best line of defense against influenza, considering the limited therapeutic options that are currently available. Now, one of the reasons that the paper focused on influenza A is that the two antigenic components, namely hemagglutinin and neuraminidase, are composed of strains of influenza A virus, namely H1N1 and H3N2, as well as influenza B virus subtypes. Now, one of the problems associated with the vaccine is it requires annual reformulation due to antigenic variation, namely mutations associated with hemagglutinin and neuraminidase. The other drawback is that vaccine strains must be selected well in advance of the start of the influenza season, up to 7 to 9 months prior to that season, due to the fact that we have to produce many doses associated with the vaccine, currently at about 1.5 billion doses worldwide. So before we get into the details of the paper, I wanted to go through the structural and genomic components of influenza A virus. So influenza A is an enveloped, single-stranded, negative-sense RNA virus with a segmented genome. And it has 8 segments, which include the primary vaccine antigen hemagglutinin and the secondary vaccine antigen neuraminidase. The other important thing to note is the RNA-dependent RNA polymerase, which is composed of 3 subunits, PB1, PB2, and PA, as well as a non-structural protein, NS, which is responsible for the export of viral RNAs from the nucleus into the cytoplasm. And finally, it includes the nucleoprotein, which encapsulates each of the viral RNAs, as well as the matrix proteins. Now, as many of you know, reassortment can cause pandemic strains, which occurs when 2 different strains of influenza affect the same cell. You can get a mixing of the different genomic segments. But as we'll talk about shortly, reassortment is also used to create vaccines. Now, one other thing that's important to note with respect to the advancements of the paper is that the current influenza vaccine manufacturing process consists of a plasmid reassortment that's used to combine genes to make what's called seed viruses. And in this case, the 2 antigenic components that change seasonally, namely the hemagglutinin and neuraminidase, come from a WHO candidate virus. But the remaining 6 genes come from the viral backbone. And that viral backbone can either come from H2N2, in the case of a live attenuated vaccine shown on the left, or H1N1 in the case of an inactivated vaccine shown on the right. Now, once the seed viruses are obtained, they're then amplified in embryonated chicken eggs. Now, there are several disadvantages of this approach. The primary one being that this multiplasmid transfection has a very low virus yield because not all the 8 plasmids will transfect every cell. In addition, there's manufacturing scalability issues, including, for example, slow replication process and a multi-step process which adds time into the manufacturing. Furthermore, the process relies on pathogenic-free chicken eggs. This is a problem if avian flu is prevalent in the chicken eggs at the time of the manufacturing of the influenza A vaccine. In addition, You can actually get egg adaptation mutations associated with the virus that can actually change the viral components. And then finally, there's biosafety issues associated with handling infectious virus. Now, the paper is going to address several of these drawbacks, as we'll discuss shortly. Okay, so next we want to talk about some of the research design details and key results. And we'll start out by talking about some of the design objectives. So one of the major design objectives in the paper is to develop a modular and inducible vaccine platform called IMD, which stands for Inducible Master Donor, to make infectious influenza A virus particles. And the idea here is that we want to integrate into the host genome of the cell line what used to be provided by the PR8 and MDV plasmids discussed earlier, which contain the 6 genomes of the virus backbone. So we want to integrate all but hemagglutinin and neuraminidase influenza genes into the host cell genome. And then later, we will transfect those IMD cell lines with HA and NA via plasmids. And then subsequently, we will induce production of virus particles by 2 chemical compounds that we'll discuss in a moment. Second objective is we want to ensure that these viral genes are inheritable and stable. So we want to make sure that they're passed on to their progeny. We want to make sure that we avoid cellular toxicity. And we also want to make sure that we can incrementally test each cell line, intermediate cell line, at each stage of the design process. So in order to create the inducible master donor cell line that can be transfected with hemagglutinin and neuraminidase at the end of the process, they created a modular staged design approach. They started with human embryonic kidney cells as the baseline. They then created an initiation gene set where the genes are associated with the RDRP complex and integrated into the genome of the human kidney cell line. This allowed mRNAs for RDRP to be created in the nucleus and transported into the cytoplasm to create the protein complex for RDRP. They then created a genome module, which allowed them to produce viral RNAs of the RDRP complex as well as the non-structural protein NS. NS allows viral RNAs to be exported from the nucleus into the cytoplasm. Then finally, they created a booster module, which consists of genes associated with the remainder of the gene set, namely the nucleoprotein and the matrix proteins. At the conclusion of the process, they have the inducible master donor cell line that can then be transfected with plasmids containing hemagglutinin and neuraminidase to form infectious virus particles. So one of the things that's important to understand is this idea of inducible promoters. So it's important to note that both polymerase 1, Pol1, and Pol2 are host cell polymerases, not viral polymerases. So Pol1 actually promotes the viral RNA and is not inducible, whereas Pol2 promotes mRNA and is inducible. And the 2 chemical compounds that they use to induce the mRNA are doxycycline, which activates what they call the TET-on promoter, and mifepristone, which activates the P-switch promoter. And the reason they use 2 different promoters is that it allowed them to get greater control over the process. So as I said before, the RNA-dependent RNA polymerase is comprised of influenza A virus PB1, PB2, and PA subunits. And one of the reasons that these are controlled is that if they're not controlled, they can actually lead to cellular toxicity. Okay, I wanted to get into some of the details of the different cell design stages. So as I mentioned earlier, the initiation gene set, which is Pol2 inducible, integrates the 3 components of RNA-dependent RNA polymerase via lentivirus vector into the host cell line genome. And these 3 subunit mRNAs are inducible by doxycycline, so they don't cause cellular toxicity in the cell line. And as you can see on the diagram on the right, where those genes sit in the overall architecture. In the second stage, we have the genome module, which is Pol1, which is not inducible. So remember, Pol1 produces viral RNAs, not mRNA. So what we want to do is we want to supplement what we previously introduced into the genome with actual genes that can produce viral RNAs. So we want to integrate PB1, PB2, and PA via leap-in transposon into the host cell line genome so we can produce viral RNAs. We also want to add the NS, non-structural protein, into that genome to allow the nuclear export of those viral RNAs for subsequent testing. And then finally, we have the booster module, which is both Pol1 uninducible and Pol2 inducible. So at this stage, we want to integrate the remainder of the genes, namely the nuclear protein and the matrix protein, via leap-in transposon, whereas the mRNA associated with those two genes are inducible by mifepressone, Pol2, and by Pol1 producing the viral RNAs, which are not inducible. So now we want to get into some of the key test results of the paper. And one of the advantages of the stage modular design is that we can actually test to make sure things are working along the way. We don't have to wait to the end of the process to make sure those inducible master donor cell lines actually contain all of the genes that we want. So the figure on the left shows that the RNA-dependent RNA polymerase, which has been those genes have been integrated into the host cell line genome, are actually producing those proteins. And those proteins, in turn, can actually replicate what otherwise would have been the HA gene introduced by a plasmid, but that's been replaced by the green fluorescent protein, so we can see the results. But the figure on the lower right shows that, in fact, that RNA-dependent RNA polymerase is actually working. Likewise, at the second stage, in the middle of the figure, we show that as a result of the genomic gene module being integrated into the host cell line, that these viral RNA expressions are actually produced and packaged into infectious virus particles, with again an HA gene being replaced by the green fluorescent protein, so you can see the results. And then finally, we show on the far right the fact that they actually produced a fully inducible influenza A virus production in two different clones of that virus particle. And again, with the HA gene replaced by the green fluorescent protein. So at each stage of this process, they can determine that things are working properly. So one of the things they noticed was that the viral RNAs produced using this approach were actually low when compared to wild-type infected cells or cells produced by egg-based production. And as you can see on the figure on the left, that the titer levels of clone 1 of the inducible master donor cell line were on the order of 10 to the 7th. But when you look at wild-type cells or cells produced by egg-based production, they're typically on the order of 10 to the 8th to 10 to the 9th PFUs per mil. So the authors investigated the reason for this discrepancy, and they performed a transcriptomic analysis for the different genes associated with influenza A. And as you can see on the figure on the right, when comparing the viral RNAs to either the complementary RNA, the cRNA, or the mRNA produced by host cell polymerase II, that depending on the gene, the titer levels are about 1 to 2 orders of magnitude low. So they performed a subsequent analysis to determine the cause of this discrepancy. So as part of this analysis, they also looked at the genes associated with the innate immune system. And in the figure on the right, they computed the fold change associated with the uninduced master donor cell line when compared to the HEK293 T cell line on the x-axis. And on the y-axis, they computed the fold change associated with the induced master donor cell line compared to the uninduced master donor cell line. And as you can see in the figure, the genes were upregulated either as a result of the integration of those genes into the host cell line or the induction of those genes. And I've highlighted 4 genes associated with interferon stimulated antiviral response. And as a result of these genes being upregulated, there's a negative impact on the virus titers. Now, the good news is that if these genes are knocked out in the host cell line, this could lead to increased viral titers and increased production of influenza A virus particles.
Hello, my name is Ravi Chandramoli, and I will be discussing the significance and broader impacts of this study. As David has earlier discussed, the researchers of the study have shown a novel technique for understanding and producing influenza A virus components in synthetic cells. What is significant about this study, though, comes from a quote from the authors in the introduction in regards to traditional virus culturing. The process, in traditional virus culturing, depends on the supply of pathogen-free chicken eggs, which could be disrupted during pandemics. Flu vaccine production in cell cultures has been shown as a better alternative as it can be improved quality control and greater surge capacity. Why is this quote significant? It acknowledges the challenges that can be faced with viral culturing and how they are addressed them in the synthetic cell lines in this study. Traditionally, culturing influenza A is a labor-intensive process that requires transfection of multiple plasmids, followed by propagation into embryonated eggs or cultured cells. This approach is time-consuming, costly, and dependent on external resources, such as pathogen-free eggs, which can limit scalability and consistency. In this study, the researchers applied synthetic biology techniques to engineer inducible synthetic cell lines capable of producing all the essential viral components, excluding the key virulence factors hemagglutinin and neuraminidase. By removing these components of virulence, the team established a genetically contained and biosafe system for generating viral particles. This innovation provides a faster, safer, and more reproducible alternative to conventional virus culture methods. It removes the need for outside materials that can be constrained, genetically minimizes the safety risks, and lowers the overall cost and materials and time, resulting in a platform that can accelerate experimentation. Although this study focuses on influenza A virus and demonstrates how faster, safer culturing can be achieved, its implications extend well beyond a single pathogen. The work illustrates how synthetic biology techniques can be harnessed to advance viral research, offering new strategies for studying virus biology, developing vaccines, and testing antiviral compounds in a controlled and reproducible manner. While molecular tools such as PCR, CRISPR-Cas systems, and next-gen sequencing have greatly improved viral detection and genetic analysis, effectively monitoring and control remain challenging because viruses mutate rapidly and adapt to their hosts, often evading immune responses. These challenges highlight the critical need for systems that enable researchers to rapidly characterize viral components and behaviors, particularly when resources are constrained or containment capacity is limited. The synthetic inducible platform developed in this study is such a foundation, a reproducible, biosafe backbone that supports rapid investigation of viral mechanisms, which can accelerate the development of vaccines or treatments. By integrating modular design and genetic control into the design of the experiments, a bridge can begin to be formed to fill the gap between diagnostics and therapeutic research. The significance and broader implications of this study have been noted. This paper also shows a paradigm shift in how researchers can approach experimental designs. Influenza A virus production requires complex, labor-intensive transfection of multiple plasmids followed by culture in eggs or cell systems. Here, the researchers replaced that process with synthetic cell lines, which have inducible control over the viral component expression. This changes the virus production from a manual, multi-step process to a programmable and reproducible system. The study also shows how biosafety practices can be engineered into experiment design. Instead of depending solely on physical containment within a laboratory, the researchers demonstrated genetic biocontainment, removing a virulence genes such as hemagglutinin and neuraminidase to prevent uncontrolled infections. This approach embeds safety directly into the experimental design, allowing for faster and safer and more controlled culturing of viral particles. Overall, this work illustrates how synthetic biology can reshape virology into a more efficient, scalable, and inherently safer field. By integrating precision genetic tools with biosafety principles, the study sets the stage for a new generation of experimental systems that advance both scientific innovation and public health preparedness.
Hello, everyone. My name is Kyle Headley. I'll be talking about the limitations of the study as well as the future directions and follow up studies for our paper. I wanted to first take a look at the limitations of the study that were identified and throughout the study and also mentioned in the study. First, one of the major challenges that was highlighted was the influenza A's large viral genome. Uh, Having such a large viral genome does make the process of genome integration difficult. Genome integration in general is always more technically difficult. Finding the right area to integrate specific genes or productive production genes, right? Um, and incorporating these multiple gene segments into the host cells specifically, can be complex and lead to insufficient viral productions, um, and can also be impact the cell's structural stability. So overall, that had a huge impact on the study. Uh, Going into the next limitation of low viral genome amplification, particularly want to mention the induced IMD cells that they showed insufficient amplification of viral RNAs. The limitation directly affects the influenza A virus productivity through the paper, which suggests that the process that was implemented is maybe incomplete or was not at its most optimal level. Um, Obviously, that's a challenging thing to troubleshoot through. That can also be, looking at into the future studies here in a little bit, but increasing the viral yield, so potentially adjusting the cellular engineering. Um, an important limitation I want to also take a look at is the leaky expression and the cell line construction of the study. The study found that there was leaky expression of the viral RNA dependent and RNA polymerase genes, even under in uninduced conditions. The unintended expression may have placed stress on cells or interfered with the precise timing of viral gene expression, which could ultimately have that effect on the reduced viral productivity. Going into that as well of how we had the low viral gene amplification. With collectively these limitations and collectively of the study, right, and it indicated that the systems still show potential, right, because the study was a success, but for these limitations, it could definitely you could definitely see future studies and future projects of trying to improve on optimizing these pathways and the viral replication efficiency as well as like the production yield. Looking at the future perspectives and follow-up studies, um, I wanted to first talk about the cellular engineering that could be utilized to increase viral yield. Uh, it's one of the I feel like if they do future studies or a group of people take this and want to continue on with the influenza A and try to enhance the cellular engineering, one strategy that we've talked about even in this class and I've done in other classes is potentially deleting or inhibiting the key antiviral response genes. Specifically in this study, the IRF7 or the OAS3 gene, by reducing the cell's ability to innate antiviral defenses, that could allow for a potential, more efficient viral replication and higher overall productivity. Additionally, maybe finding a more optimal section of the genome to in uh, place in the selected genes that they're looking for. Next, I want to take a look at the improved integration strategies, right? So, how I previously had stated, but you could definitely include using like the multi-module integration approaches, like the AAV systems that we've discussed. The AAV could significantly shorten the cell line construction time and potentially enhance the stability and yield of the viral genome integration. Lastly, I wanted to talk about how researchers could explore alternative host systems and promoter designs. So, for example, they could potentially use different host cells. The host cells that they utilize maybe weren't as efficient as they had hoped. So you could maybe try host cells such as like the Vero cells or the Medine Darby canine kidney cells. These could potentially uh, provide a more suitable environment for the viral production that is needed for this project. There'd obviously need to be more research done to determine whether or not these cells would be sufficient enough for its entire purpose. Um, Additionally, integrating a tighter promoter system could help reduce the leaky expression and offer better control over viral gene induction.
My name is James Wynn, and I'm going to expand on materials and methods. This slide shows the stepwise construction of the inducible master donor, or IMD, cell line. They began by using lentiviral vectors to transfect the initial polymerase gene set, PB1, PB2, and PA, into HEK293T cells. These were placed under an inducible TET-on promoter so the polymerase complex could be activated only when needed. Next, they used the leap-in transposon system to integrate the genomic and booster modules, which together include the remaining viral genes required for replication and packaging. To enable tight inducible control, two transactivators were used. RTA, which responds to doxycycline for TET-on activation, and GSTA, which responds to mifepristone through the P-switch system. Each integration step was confirmed using antibiotic resistance markers, puromycin and blastocytin, for the initiation set of the inducible RDRP, hygromycin for the genome module, and zeocin for the booster module. They performed 2 rounds of lentiviral selection for RID-RP and single-cell cloning after the final integration to isolate stable lines. The resulting IMD cells carry all influenza A virus genes except HA and NA, which can later be supplied by plasmid transfection to trigger full virus production. The system was designed around 3 modular components: initiation, genome, and booster, to allow precise and independent control over replication and assembly. The initiation module, delivered by lentivirus, carries PB2, PB1, and PA, the RNA polymerase genes. Because these genes are cytotoxic when expressed continuously, they're controlled by the TET-on promoter, activated by doxycycline through the RTA transactivator. The leap-in transposon system was used for both the genome and booster modules because it can stably integrate much larger DNA fragments than lentivirus, ideal for multi-gene influenza segments that must remain intact and heritable. Finally, the gene switch system, driven by GSTA and responsive to mifepristone, was used to fine-tune the expression of NP and M genes during packaging. Each vector and promoter thus served a defined purpose. Lentivirus provided efficiency and safety, leap-in assured stability and payload capacity, and the inducible promoters gave temporal precision and protection against cytotoxicity. After integration, they confirmed that the inserted genes were both functional and tightly regulated. First, they tested the RNA-dependent RNA polymerase complex, PB2, PB1, and PA, using a delta HA-GFP reporter assay. In this system, the HA gene is replaced with GFP, so fluorescence occurs only when the viral polymerase is active. GFP signal appeared exclusively after doxycycline induction, confirming that the TET-on system successfully activated RD-RP function without background expression. Next, virus rescue experiments were performed using the single-cycle Delta HA virus in the PR8 strain. Infectious particles were produced only under induced conditions. Critically, no virus formed in the uninduced cells, demonstrating that promoter leakiness was minimal and that the system prevents unwanted cytotoxic stress. Together, these results confirmed that the IMD cell line's inducible modules operate exactly as intended, silent until triggered, and fully functional when activated. One of the key strengths of this design is its incremental testable build process. In stage 1, we established the IRD-RP line, which contains only the polymerase genes PB2, PB1, and PA under TET-on control. Induction with doxycycline confirmed active polymerase function. In stage 2, the inducible RD-RP4V line was created by adding 4 influenza genome segments, PB2, PB1, PA, and NS, through LeapN integration. Induction tests confirmed both vRNA and mRNA transcription. In stage 3, we introduced the booster module, encoding NP and M, to generate the final IMD line. When HA and NA were later supplied by plasmid transfection, these cells were assembled and released infectious influenza A particles. Validating each stage independently ensured integration, inheritance, and inducible control were maintained throughout the build, ultimately producing a fully programmable virus production platform. Broader significance. The IMD platform replaces transient multiplasmid transfection with a stable, genome-encoded, and inducible system for influenza virus production. This shift enables automated, safer, and more scalable vaccine manufacturing, where viral replication can be toggled on or off with chemical inducers instead of repetitive plasmid delivery. The modular inducible framework also extends beyond influenza. Similar strategies are now being applied to recombinant AAV and other viral vectors, pointing toward universal packaging cell lines for gene therapy and vaccine application. In short, this work demonstrates a synthetic biology approach to programmable biomanufacturing, a proof of concept for the next generation of modular, controllable virus production systems that could redefine how vaccines and viral therapeutics are made.